So this is lecture 6 in the series of 8. Over the past several lectures, we've been covering some of the fundamental aspects of clinical trial design and analysis. We started out by talking about why we do clinical trials and the challenges in defining the question and how to measure the question. We covered some of the basic trial designs. And in the last lecture, we focused on the aspects of estimating the size of a study. As you recall, the size of a study depends on the particular outcome variable that you're measuring, the differences that you want to detect, the variability of that outcome, the rate of a false positive, as well as the sensitivity of the study. There are many other factors, but those are some of the key ones that take into account how large a study to be. As we discussed, it's important for a study for ethical and scientific and economic reasons to be sized properly. Today, we're going to talk about another topic that's monitoring a trial for evidence of safety issues as well as benefit. I presented here some references which cover this topic in more detail. There are particular textbooks now that I would refer you to that are specifically devoted to this topic, as well as chapters in other textbooks. Monitoring is not a new topic, but as you'll hear later, it's a topic which has gained renewed interest. It actually has a 35 or almost 40-year history. In the 1960s, a committee which has been labeled the Greenberg Report looked at how trials should be organized and monitored. As you'll see, this was implemented in a study called the Coronary Drug Project, and later guidelines have been issued by both the FDA as well as the NIH. We'll look into those briefly. So the Greenberg Report was commissioned in the mid-1960s by the National Heart, Lung, and Blood Institute to examine how a trial should be conducted, what the structure should be, what the issues should be. And among the recommendations was that every trial should develop a mechanism to terminate a trial early. If, for example, the question has already been answered, there's no need to continue. Or, to the contrary, if the trial can't ever achieve its goals. There may be some unusual circumstances, like another trial has already answered the question. Or it may be that the entire basis of the study is no longer relevant. So a sponsor, according to the recommendations, should decide to terminate a trial only if they have advice of an external committee. This independent sort of checks and balances was a critical component to the recommendation. The first trial to actually implement the Greenberg Report was really the Coronary Drug Project. It had an advisory board made up of senior investigators, external experts, NIH staff, and initially they reviewed the interim data. There was a statistical center or a data coordinating center that pulled the data together and provided interim analyses. After they'd been operational for a few years, they realized that there was so much for the policy advisory to do that they formed a subcommittee, which they called the Safety Monitoring Committee, to examine the accumulating data for benefit or harm. I've depicted here in this figure the overall organizational structure of that early trial. The funding agency, which was the NIH, the steering committee, which was the academic experts, plus some NIH experts that sort of provide the guidance and direction. A lot of clinics, of course, are involved, which is where the patients come from. Central laboratories contribute to the database, clinical laboratories, electrocardiogram laboratories, and so forth. And many working committees which were involved with recruitment issues, entry criteria issues, and so forth. All of this data is put together in the Data Coordinating Center, who has a reporting responsibility to this advisory board, and in this case, particularly the Safety Monitoring Board. That structure worked very well for the Coronary Drug Project, so well that it became sort of the prototype model for all of the National Lung, Heart, and Blood Institute, as well as many other institutes at NIH. I've simplified the diagram here because often the Policy Advisory Board is not one that is used as a concept much anymore, but the Data Monitoring Committee concept is. Otherwise, all the pieces from the original Coronary Drug Project remain intact. As I've indicated here, this idea of a clinical trial structure not only became the standard for the National Heart, Lung, and Blood Institute, but many other institutes over time, including the National Eye Institute, the National Institutes of Diabetes, Digestive and Kidney Disease, National Cancer Institute, and so forth. In the early 1990s, industry, the pharmaceutical industry in particular, had a large pipeline of trials that needed to be tested in the Phase 3 definitive trial design. It was a time that NIH funding was becoming quite tight. So the cardiologists who had been pioneering this kind of a clinical trial idea teamed up with industry and began to use this model for trials that were being sponsored by industry. The FDA, which of course regulates many of the industry studies, didn't have much to say about the structure of data monitoring and data monitoring committees. The guidelines in 1989 are very brief. However, over the 1990s, an international effort called the International Conference on Harmonization had several initiatives which included two sections in the ICH E9 that referred specifically to interim analysis and independent data monitoring committees. So now we have in the mid-1990s comment theory from both the FDA and the NIH about how trials should be conducted and how they should be monitored. Well, when do you need independent data monitoring committees? Up until the year 2000, this was something that was reserved for very special trials, those sponsored by the NIH primarily, and a few industry trials that were academic-led. But in the year 2000, an event took place. There was a death of a patient in a gene transfer trial. This brought a lot of attention, public attention, political attention, and led to Secretary of Health and Human Services, Donna Shalala, to issue a requirement that all NIH and FDA trials must have a monitoring plan, a formal monitoring plan, and for some trials, they may require a formal data monitoring committee. The NIH already had drafted a policy to sort of formalize what they'd been doing for 30 years, that all sponsored trials had to have a monitoring system, and all phase 3 trials, which we've talked about, had to have a data monitoring committee. The FDA drafted guidelines shortly thereafter for themselves. Those draft guidelines came out in the year 2001, but have been formalized and finalized in the year 2006. Because of that gene transfer issue and the mandate that Donna Shalala issued, policy of data monitoring became not optional but required, which is why we spend the time in these lectures to cover some of the basic fundamentals. Let's look at this more carefully. In phase 1 trials, as we talked about a little bit, you're looking at the first-in-man studies, and you're trying to figure out the dose. Most of those designs is you escalate from a low dose to a high dose. You do not escalate until the previous dose was satisfactory in terms of toxicity. So there's a built-in monitoring process, usually within the local institution. Phase 2 trials, there's also a monitoring process built in because typically you put some patients on the study, you decide whether there's any activity of the agent or the device as designed, and if there is, you move on to a second stage and have a somewhat larger study to refine the toxicity profile and convince yourself that the activity you desire is there. So there's a built-in data monitoring process. The phase 3 and sometimes called phase 4, the post-approval studies, are the most frequent users of the Independent Data Monitoring Committee. Generally speaking, the structure of the monitoring needs depends on the risk in the study. The FDA, for example, doesn't require data monitoring committees for all trials, but clearly suggests a need for an independent monitoring committee for pivotal phase 3 trials or trials which have a mortality or the potential for an irreversible morbidity outcome. The NIH model, as I described earlier, was modified slightly in this figure, to include the regulatory agencies, in addition to the industry sponsor, as well as to divide the coordinating center, where the data used to go and the analysis came from, into two pieces. A data management center, which could be the sponsor itself or some contract research organization or CRO, and an independent statistical center, which provides the analysis both on an interim basis as well as the final analysis. This has become quite a common model now, generated in part by the FDA regulatory agencies wanting the independence. And secondly, many journals now want independent analysis of industry-sponsored studies. So this model, in fact, serves multiple purposes and is actually quite useful. The Data Monitoring Committee is a special committee that has responsibilities and relationships to a number of groups. First and foremost are the patients. Their primary responsibility is to try to protect patients from being in trials where there's convincing evidence of benefit or harm. Also, they have responsibility to investigators who have given them this responsibility on their behalf. Obviously, the sponsor of the trial is the ultimate group that's responsible for a trial, and so the monitoring committee reports their activity to the sponsor as well. A less well-understood relationship is to the local institution review boards, who depend on the data monitoring committee to do the monitoring of safety, particular safety, on their behalf, although this is not always well understood. And finally, there's responsibilities to the regulatory agencies who depend on this body, this data monitoring committee, to again monitor safety on an ongoing basis. So in summary, the data monitoring committee has relationships to several institutions or groups of people, and these must be taken into consideration as they go about their business. Regulatory agencies, for example, might be concerned about a specific issue, but it's generally been agreed upon that the FDA, the Food and Drug Administration, or other regulatory agencies themselves should not participate in the closed sessions of the data monitoring committee work. The main reason is it's hard to participate in the decisions and then have to later give an independent regulatory review. However, the FDA needs to be briefed about the decisions that the monitoring committee makes. What does a data monitoring committee actually review? Well, there's several factors, several components. I've listed a few here in this slide. First of all, they want to review and monitor recruitment. If you're not recruiting patients fast enough that you'll ever meet your target, then the question comes up as to why are you continuing if you will never accrue the patients needed to answer the question, both an economic, scientific, and ethical issue. They'll also examine carefully the baseline variables, that is, what does the patient profile look like? For a variety of reasons. They want to understand whether the patients in the trial really eligible, and they'll also want to check on the comparability of the two arms of the study, the control arm and the new experimental arm. Obviously, they will look at outcome measures to assess benefit, both the primary and the secondary outcomes. And for safety, they'll look at both serious toxicity and other adverse events. To examine the quality of the trial, they will look at the compliance of the patients to the therapy as specified in the protocol. And they may very well examine specific subgroups to see whether the treatment is particularly beneficial or particularly harmful in some class of patients. This is a small list, but the key list of what a data monitoring committee might review. After this review, they can make recommendations to either continue the trial with no modifications and wait for a new update on the data. They may make a decision or recommendation to modify the protocol because something isn't quite according to the plan. In some instances, they may decide to recommend termination of the trial. A design modification might be, for example, to modify the entry criteria to exclude some patients or to open it up to a wider range of patients. They might decide that the dose isn't high enough to get the activity that's hoped for. Under special circumstances, they may recommend sample size adjustments or a change in the frequency of measurements. In general, monitoring committees do not like to make design modifications, but in some cases it may be necessary. A monitoring committee could terminate a trial early because there was established serious toxicity, or the benefit was convincing and already established, there was no need to continue the trial further. On occasion, trials may be stopped for futility because the emerging trend is either of no interest or it's not sufficiently beneficial that you'll ever achieve statistical significance at the end of the trial. Fourth, hopefully not too often, the design or the logistical issues may be too serious to fix, in which case you need to stop and possibly start over. But I'd like to emphasize that the decision-making process to continue, to stop, to modify is very complicated. It depends on a variety of factors. Are you meeting your recruitment goals? Do you have the kind of patients risk that you need? Are they complying to the therapy as hoped for? What are the trends in the primary and secondary outcomes? What does the safety profile look like? Is there internal consistency across the primary and secondary outcomes? Is there internal consistency across subgroups? Are the data that you're seeing consistent with external data, or is it quite unexpected? Ultimately, what's the benefit versus the risk? And there's an interesting ethical question about the patients in the study versus patients who might come into the study, and even beyond that, future patients. You also have to examine what would the clinical or public impact be of stopping the trial at this point in time. If you stop a trial because you believe it's beneficial, but the benefit is of marginal value and has no clinical or public impact, then you've not accomplished anything by stopping early. Finally, I've listed statistical issues, and we'll examine those in just a minute. It's important to emphasize that the Data Monitoring Committee typically makes recommendations, not decisions. The reason for that is that they have a lot of people to report to. the investigators, the sponsor, and in some occasions the regulatory agencies. So they can make recommendations, and they can certainly debrief or provide the steering committee or executive committee of the trial or the sponsor, or even if the regulatory agencies want to be briefed. Experience is such, though, that rarely are data monitoring committee recommendations rejected. Over time, a particular format for data monitoring committee meetings has emerged. An open session is a session where investigators, sponsors, other people who have a vested interest in the trial may be present to discuss the progress of the trial. The data are blinded, as it's not discussed as treatment versus control, but overall merged data may be examined if necessary. But it's a chance, an opportunity for an open exchange. Once that has taken place, the data monitoring committee goes into what's called a closed session, where the data are examined by treatment versus control. Present is the data management center and the statistical analysis center. One, because they collect the data. Two, because they analyze the data. It's not regarded as good practice for the sponsor to be present, because the sponsor is in a position of making operational decisions and changes. Making those changes with the knowledge of emerging trends is viewed as an opportunity to introduce bias into the study. So in general, for industry-sponsored trials, this is not recommended. For NIH-sponsored trials, there is still some debate. Then, after the data have been examined carefully, the data monitoring committee may go into an executive session where just they alone have a chance to discuss amongst themselves their feelings and recommendations. Once they've come to their recommendations, there'll be a final session with at least some representation of the study, the sponsor, an executive committee, for example, that debriefs them as to the decisions of the monitoring committee or recommendations of the monitoring committee, and to fill in any details that they need to know about those recommendations. Membership, who do you need on a committee? Well, whether your trial is a large one or a local institutional study, most of the principles I've talked about these days would be necessary. The number of people depends on the complexity and the size of your study, but you probably need some level of clinical expertise, some level of basic science expertise, someone who understands overall clinical trial methodology and biostatistics, in many cases the epidemiology of the disease, and someone who represents the medical ethics. Unlikely that would be one person, but usually 2 to 3 people could be for moderate-sized studies. If the trial is large and complex, you may need as many as 5 or 6 to 7 people. It's helpful to have some regulatory expertise, but as I said earlier, probably regulatory people do not want to be on the committee because later they may have to give independent judgment. Experience is always helpful, especially for the chair. It's understood, in fact, sometimes it's formally written, that the interim data that the committee will review is confidential. It remains confidential until you come to an agreement that the trial has established benefit or harm or futility. On a rare occasion, a monitoring committee may release specific data limited to the interim data for some safety issue, for example. It may be that a drug or a device has some safety issue that they want to alert the patients and the physicians to in order to avoid those issues in the trial. In the hormone replacement therapy trials, it was understood or discovered that there was a clotting problem. In both trials of that HRT, the data monitoring committees alerted patients to the clotting problems. But in general, members must not share interim data with anyone outside the data monitoring committee. The reason is that leaks can affect patient recruitment, protocol compliance, can bias the outcome assessment, and perhaps could even affect the support for the trial. The goal is to get the answer. That's why you started the trial, and you don't want to do anything that would jeopardize getting the answer on behalf of the patients as well as the sponsor and the investigators. All of this requires that you have in place what I might call an online data management and analysis system. No monitoring committee wants to make decisions on old or incomplete data. There are examples, which I've alluded to here, that the data are delayed in getting into the database and into the analysis, or the events that are being examined are not verified. You may be working off of old data and could, in fact, make decisions which you would later regret. The nocturnal oxygen therapy trial and the ACTG 019 trial are examples of that. The nocturnal oxygen therapy trial had a data delay from a couple of sites and could have come to the conclusion that the results were effective only in one subgroup and not in others. the delay was discovered, and when the database was updated, that decision would no longer have been valid. Similar example in the AIDS clinical trial group, trial 019. The important message is you must have your data management system in place, ready to roll at the beginning of the trial, because you don't know when trouble will start, as it occurred in the cardiac arrhythmia suppression trial, or CAST, where very quickly drugs designed to suppress arrhythmias turned out to cause an increased rate of mortality, or you may in fact have data coming in slowly and make the wrong decision. You can't have everything coming in at once, and so you need to focus your energy on key variables, primary, secondary, and perhaps serious adverse events. We talk about levels of independence, especially these days with all the sensitivity by journals, political, and public authorities. Here I've listed one concept of levels of independence, from being a totally in-house within the sponsor operation to one where there's internal data management, but external statistical analysis and an external data monitoring committee. The next level of independence would be an internal data management system, but an external statistical analysis center and an external data monitoring committee. And finally, the most independent would be if the entire data management operation is external to the sponsor, the data is analyzed externally and reviewed externally. The last two I've highlighted in blue because they're probably the most desirable. Certainly the third bullet, that level of independence, is by far the most common and also the most desirable. So again, this industry-modified NIH model, whether it's for NIH or industry, the components are identified, and after 20 years of experience, this model works pretty well. And to emphasize, whether it's a large study, multi-center, with a large budget, or whether it's your own trial within your own institution or your own department, the components that are represented here need to be considered and make sure that you have them covered in some way. So in summary, the data monitoring committee process has a long history of success going back into the late 1960s on NIH-sponsored trials. It was adapted successfully for industry. As I've indicated, the Statistical Center, the Data Monitoring Committee, Statistical Analysis Center are critical components of this model. The most important, I believe, is the independence of the monitoring committee. That's essential. And the best way to achieve these goals, in addition, is to have an external statistical analysis center and an external data monitoring committee. Let's look a little more carefully at the data monitoring process and the statistical issues that go along with it. I've given a reference, if you would wish to read more detail, about one particular trial, the beta blocker heart attack trial, was published in 1984. As I've indicated, there are many decision factors, and I want to focus now on the statistical issues, in particular the ones of repeatedly testing data. Beta blocker heart attack trial, in brief, was a trial conducted in post-heart attack patients. Mortality was the outcome. It was randomized, double-blind, placebo-controlled, with an extended follow-up. 3,837 men and women were randomized that had a heart attack 5 to 21 days prior to entry. The trial compared propranolol versus placebo. What I've listed here is the outcome, the interim results for the primary outcome variable mortality as it occurred in calendar time, beginning in May of 1979 through October of 1981. The trial was scheduled to go almost another 6 to 8 months. In the May meeting of the Data Monitoring Committee, it was observed that there was 22 deaths on propranolol and 34 on placebo. The test statistic for that comparison, which we will not have time to discuss here, but it is on the standardized statistical scale, the Z-scale, is a value of 1.68. That Z-value does not support a p-value less than 0.05. October of 1979, there were additional deaths, 29 versus 48. Now the Z-value is increased to 2.24. The trial continues on to October of 1981. There was 135 deaths versus 183, and the Z-value is 2.82. There's an asterisk there indicating that's the date at which the monitoring committee recommended termination. Not listed is that the p-values associated with those Z-values would have been less than 0.05 from the October 1979 meeting onward. Why didn't the trial stop when the p-value was first less than 0.05? Let's examine that. What I've shown here is the mortality curves as observed on October 1st of 1981 with the Z-value for the log rank statistic of 2.82. As you can see, the mortality rate on placebo is higher from the beginning, essentially, than those patients treated with propranolol. While the initial review of the data is done for ethical, scientific, and financial reasons, as I indicated, But the process of repeatedly looking at data causes a statistical problem that we have to address. What I've shown here to illustrate why is another trial. In fact, it's the Coronary Drug Project, the first one that implemented the Greenberg report. As shown here are mortality results for the clofibrilate-treated patients versus the placebo-treated patients. This is a trial in post-heart attack patients, men in this case. And as you can see, the mortality curves are essentially identical. However, on this graph, we plotted the data as it came in calendar time. On the x-axis is time in months of the study. On the vertical axis is the test statistic on the standardized scale, going from -3 to +3. In this case, a positive value means that the placebo is superior. A negative value means that the drug clofibrilate is superior. As you will notice, the data vary over time. On 4 occasions, the data approach a p-value of less than 0.05. That is, the z-value was nearly -2 or slightly larger. Yet, remember, by the end of the trial, there was literally no difference in the mortality between the clofibrilate-treated patients and the placebo-treated patients. This is the dilemma of every monitoring committee. When is the signal noise and when is it real? This has been examined very formally by statisticians, and if you use the same criteria, a standardized statistic of 1.96, which is equivalent to a p-value less than 0.05, if you use that rule every time you examine this data, you no longer have a type 1 error of 5%. It increases with each look. If you look twice, that type 1 error or false positive error would occur with probability of 0.08. 5 interim analysis, which would be quite typical, would increase to 0.14. And 10 analysis would increase to 0.20. That is, the more you look at the data, the greater the chance of finding something just by chance. So we have to be careful in interpretation of these interim results. We have some statistical methods to help us, fortunately. I want to examine one of them briefly. These methods are the group sequential method, another method which is called conditional power, and a third method which has a Bayesian approach. I like to point out that the first 2 of these methods actually can be seen being applied in that Coronary Drug Project, that very early trial. So again, not new, we've just refined the methodology. Let me give you some overview of how these methods work. There's some references listed here, but the basic idea is to compute your summary statistic every so often when the data have accumulated and the data monitoring committee is going to meet. These methods all are going to compare the statistic, the summary statistic, to some critical value, conservative critical value, such that your overall false positive rate for the trial remains at 0.05 or whatever other value level you decide to set it at. I've graphed here some of the methods that are commonly used. There are 3 methods referred to as the Pocock, the O'Brien-Fleming, and the Peto. Let me examine these quickly in order. The open circles represent the method that Stuart Pocock proposed, which is to increase the critical value, the benchmark, from 1.96, in this case, to 2.413. You raise the bar, and you have to exceed that to claim benefit, or in the opposite direction to claim harm. The next graph up, which is the open triangles, is a method that Richard Peto and colleagues proposed, which says let's raise the bar even more, but then when we get to the end, we'll go back to the standard test statistic of 1.96. So he suggested raising the bar all the way to a z-value of 3 or larger. Very conservative, unlikely you're going to reject the null hypothesis of no difference easily. And because you're so conservative, you don't have to pay any additional penalties at the end of the trial and by demanding an increased critical value. Notice that the Pocock boundary, the open circles, requires a larger critical value at the end than Peto method. The method that's probably most popular, or at least among the most popular, is the one that is represented by the closed circles, which is referred to as the O'Brien-Fleming boundary. This boundary starts out very conservative, relaxes the criteria as you go, and at the end requires a sample size, a z-value of almost the same magnitude as the Peto and conventional standards. I'll talk a little bit later about the benefits of that, but in general, this boundary reflects how monitoring committees think. Early on, they want to be very careful. The criteria is very conservative. As the trial continues, they gain confidence, they get more data, the criteria relax. At the end of the trial, the critical value for deciding success or not success is just about the same as if you didn't have any monitoring process at all. So these are 3 examples of how you can be conservative Each of them protect the false positive rate at the 0.05 level. How do those boundaries come about? Well, I don't want to go into the mathematics of this, but the O'Brien and Fleming boundary, for example, has a particular form. There's some critical value, which I've labeled Z, OBF, to represent the O'Brien and Fleming boundary, and it's modified as the trial goes on. Capital K would be the number of analysis you plan to take. The value I is the trial analysis at which you're at, the first, the second, the third, the fourth, the fifth, in the example I plotted earlier. So you notice you have this square root function, which is why it decreases in a nonlinear fashion. That's the form of the boundary. For example, if you were to have a overall type 1 error of 0.05 and plan for 5 analyses, that critical value at the end of the trial will be 2.04, not 1.96. So I said this is the boundary that is quite often used, but there's a modification coming up, which I'll share with you in just a minute. The reason that this is important for the O'Brien-Fleming boundary and why it has its popularity is not only does it reflect how monitoring committees think, but I've written down here how you might compute the sample size, which we discussed a lecture ago. If you were to compute the sample size with a 5% type 1 error, a power of 90%, and you're looking for a half a standard deviation difference, I've written down the formula and the details of what the sample size would look like. 168 patients for the two arms combined. If you were to use the O'Brien and Fleming boundary, instead of having a value of 1.96 in the formula, the Z sub alpha, you would actually use 2.04. Well, you can tell that that would have a modest effect on the sample size. In fact, if you go through the calculation, the sample size is not 168, but 170. Well, that has a big advantage if you're going to impose the monitoring process, and it doesn't cost you much in terms of number of patients or dollars spent. Then everyone is happy that this process hasn't cost the trial in terms of patients or money, but yet you've met your ethical obligation to monitor the trial in a sensible way. I've just listed here to show that the critical values will change slightly depending on how many looks you are taking at the data. For O'Brien and Fleming, for example, the critical value of the ZOBF column starts out 1.96. That's if you only looked at data once. That's standard. Twice, the critical value raised from 1.96 to 1.98. And as shown in my example, if you looked at it 5 times, it would be 2.04. I'm not going to discuss the other 2 columns in today's lecture. But if I look over at the Pocock set of boundaries, there the Z sub P is the critical value for the Pocock boundary. And notice that for, again, one analysis, it's 1.96. But if you were to plan on computing the analysis 5 times, which is in the graph, that Z value would have to be 2.413. If you think back to the sample size calculation, what you'd have to plug in is not 1.96, not 2.04, but 2.41. Obviously, that will have a much more substantial impact on the sample size. So while the Pocock boundary accomplishes the goal of keeping the overall false positive rate for your trial at 5%, it does so at a cost, which is not always a desirable property. Yet, it does allow one to terminate a trial early for harm or benefit because the criteria are not as stringent as, say, as in the O'Brien-Fleming boundary. So it has some advantages as well as disadvantages. Again, I've plotted these graphs now that we've talked about how they arise. So you can see that the O'Brien-Fleming boundary, as I said, starts out the most conservative and winds up almost in the same place as if you not monitor the trial at all or consistent with the Peto boundary. Going back to the beta blocker heart attack trial, what I plotted here is that data that I showed you earlier on the O'Brien-Fleming boundary. The dashed line represents what I would call the naive approach. It's using the same critical value 1.96 for everything, which would certainly inflate your false positive rate. But you can see as the data emerged over calendar time, it eventually crossed the O'Brien-Fleming boundary with a value of 2.82. I should point out, however, that it wasn't just the fact that the statistic crossed the O'Brien-Fleming boundary that the trial was terminated. All those issues about compliance, comparability, internal consistency, external consistency were examined in great detail before the decision to recommend early termination for benefit was made. Now a time for a brief commercial. After the beta blocker heart attack trial was completed, a colleague of mine, Gordon Land, and I, who worked on that study, realized that we had made a major step forward in applying these new methods to a major trial, but there's some limitations that bothered us. One is we were required to specify the number of analyses in advance that we plan to do. Monitoring committees never know exactly how many analyses they're going to do because it depends on what happens. The second requirement, which I didn't elaborate on, is it required that the spacing of information, number of patients or number of events, was equal between each time the monitoring committee met. Both those assumptions are not realistic and rarely ever met. So we asked the question, could we come up with an alternative procedure that had many of the benefits of these earlier methods, but got rid of these two requirements? And in fact, largely due to Gordon Land's proposal, we were able to do that. We did that by specifying what we call a spending function, an alpha star of t, which defines the rate at which the type 1 error is used over the course of the trial. T is proportional to the information, the number of patients, the number of events that you've observed so far. So you might think that t, the information fraction, goes from 0 to 1 at alpha of 0. You've spent no type 1 error, you haven't done anything. And at the end of the trial, alpha star 1, when the trial is over, you have used up all the alpha, the type 1 error that you've allocated. In between, you're going to use up some of it. As I said, information time can be expressed as the proportion of observations you have observed to date, which I've labeled as little n, the number of observed, over the capital N, the number you plan for, or it can be a proportion of the number of events, say, for example, the number of deaths you've observed, over the number of deaths you expected to observe based on your design, little d over capital D. If you go back to the graphs of the three sequential boundaries, I've listed two of them here and indicated the idea of spending alpha. Let's just examine the O'Brien-Fleming, the curve boundary, for a minute. At the first analysis, there's an arrow that's pointed above the first point on that graph. That represents the probability of crossing that boundary, starting at 0 at the beginning of the trial, which would be quite small. That probability is the probability of making under the null hypothesis, a type 1 error. If you didn't cross that boundary, you'd continue on to the second interim analysis. The second arrow represents the probability of exceeding that boundary under the null hypothesis of no difference, given that you didn't cross in the previous analysis. The first arrow plus the second arrow now represent the total type 1 error you've taken so far, the chance of claiming a difference when there isn't any under the null hypothesis. The third arrow represents the additional probability of crossing the boundary under the null hypothesis, given that you didn't cross in the first two, and so forth and so on. If you add up the probability that each of those arrows represent, you'll get alpha. The straight line, of course, is the same idea for the Pocock boundary. We can compute those probabilities. We won't discuss the details here, but the references I listed would give you those details. If you plot those probabilities, you get the following graph. Again, the horizontal axis is on information time scale from 0 to 1, and the alpha level is on the y-axis, the horizontal axis. You notice that the O'Brien-Fleming boundary uses up very little alpha, or type 1 error, at the beginning because it's so conservative. Very unlikely to cross that boundary in the first analysis under the null hypothesis. It eventually uses up all the type 1 error. The Pocock boundary, on the other hand, provides you an easier opportunity to cross that boundary early on under the null hypothesis, and thus uses a little bit more of the type 1 error than the O'Brien-Fleming boundary. Well, I've completed the graphs with those probabilities, and as you can see, there's different rates at which we use up the alpha. That was the idea of the alpha spending function. What does that do for you? Why is this such a step forward? Here I've plotted the O'Brien-Fleming boundary spending rate, and imagine that we are examining our data at some time t sub 1. We have that fraction of information in. If I extrapolate upward and hit the boundary for alpha star of t1, that will tell me what the critical value should be at that point in time. Formulas below suggest how I get that first value, c sub 1. The details are not directly computed, but require a sophisticated software package, which actually you can get from my website, or there are commercial packages available. So now after your first analysis, you know what the critical value should be. Imagine that you did not cross that critical value and you continued on to some time, information time t sub 2. You now have more information, you're not done yet. What would the critical value be? Well, as I've tried to indicate in the diagram, at time t sub 2, you now have another alpha star of t that will tell you what the second critical value should be. So I haven't specified in advance when I'm going to look. They're not equally spaced necessarily, and I can look as often as I need to. And yet I can control my type 1 error for the probability of a false positive. Well, there's a lot of complicated mathematics and statistics behind this diagram. We are able to mimic the O'Brien-Fleming boundary by the formula in line 1. We can mimic the Pocock boundary in line 2, and we can come up with other kinds of alpha spending functions. The table just suggests that the comparison of the values under one methodology versus another, and they're very close to representing the boundaries that we were looking at previously. Which boundary you choose is a matter of choice. As I said, each boundary has some advantages and disadvantages. What works best will be decided based on the needs of the trial. But in general, the O'Brien-Fleming boundary type, we say, is the most popular because it has this feature of being very conservative early and relaxing the criteria as you go to be almost the same as a one analysis critical value, thus requiring a trivial or no increase in sample size. Let's examine this idea in the context of a trial. The cardiac arrhythmia suppression trial was published in 1989 in New England Journal of Medicine. It addressed the question that cardiac arrhythmias are associated with increased risk of sudden death. A new class of drugs were developed that suppress these arrhythmias. Enconide and flecainide were among them. The CAST trial was designed to test the effect on sudden death. I've listed here 2 examinations of the data that the Data Monitoring Committee had an opportunity to evaluate. The first was on September 1st of 1988. The second was on the 30th of March of 1989. I've listed the number of events, each of those meetings, as well as the log rank or the test statistic, and the boundary values that the sequential methods provided us. In the first analysis, the z-value was a minus 2.82, representing a rather substantial difference in the mortality effect, and in fact, in the wrong direction. That is, you'll notice that the drugs have the higher mortality rate. The test statistic, even though large, did not fall into the rejection region for either benefit or harm, and thus the trial continued. By March, however, of 1989, the results had continued to get worse for the drugs, 9 deaths versus 33 deaths, and z-value, the log rank statistic of minus 3.22, which does exceed the boundaries as presented. The trial at that point was terminated after a lot of things were checked, according to the same list of compliance, comparability, internal consistency, and so forth. However, a footnote is probably worth mentioning. This was not the direction that was expected for this therapy. It was contrary to conventional wisdom and contrary to practice. These drugs were being used widely off-label because there were lots of patients who were being presented with a cardiac arrhythmia. The second footnote is that when the first meeting took place in 1988, the monitoring committee was blinded as to which was drug and which was placebo. So seeing 5 deaths versus 22, all of them probably made the assumption that the drug was working as expected, not realizing that they were in the wrong direction. A fundamental lesson from this trial is that the monitoring committee is one group of people who should not be blinded as to treatment and control, because you just never know when things are going to go in the direction you expect, or you have a surprise, as it was in this case. I've plotted here the sequential boundaries for this trial using the spending functions, as described earlier. Again, the x-axis has the information fraction from 0 to 100%, 1.0, and the y-axis is the standardized test statistic. What's important to notice is how quickly these results emerge from a z-value of 0 at the beginning, when there's no information, to a z-value of minus 2.82 and then to minus 3.22. Very quickly. Less than 20% of the trial was completed in terms of events. Several points or lessons to be learned. One is you must be prepared from the beginning to react. 2 is you have to have your data management system in place so that you can act if benefit or harm emerge very quickly. And 3, you need to have a plan in place to help you assess the trends as they emerge. An issue which I won't go into in much detail, but is something you should be aware of, is if a trial is terminated early, it's probably an overestimate of the beneficial effects or the harmful effects, because it could be a random swing that pushed you over. So there are methods available that can adjust the estimate of benefit or harm if you've crossed the boundaries. And then the naive estimates are typically an overestimate and need to be adjusted. We can even put confidence intervals around our adjusted values if we want to. And I've listed at least a couple of references for your convenience. We can also use the idea of confidence intervals to monitor the trial. This is a busy slide, but the important part is we can compute a confidence interval in the usual fashion, with one exception. We adjust the coefficient. Normally, you would think of a confidence interval as the estimate plus or minus 1.96 standard errors. Well, we're going to do the same thing, except, as I indicated, we have a theta sub L and a theta sub U, the lower limit and the upper limit, which is the estimate, the theta hat, plus or minus the standard error modified by the lower boundary or the upper boundary of our sequential plan. The effect of that is it broadens, it widens, it lengthens the confidence interval. And we can modify our plan as we go through time depending on the sequential boundaries we've used. Again, I've given some references. But if the confidence interval excludes 0, then we know that we have rejected the null hypothesis. But it still includes 0, then we need to continue on until we have more evidence. I plotted here an imaginary realization of a trial. I've given 3 data points, the estimate and its confidence interval. And you notice that confidence intervals for the first 2 examinations don't exclude delta, the difference we were interested in, but the third one does. So if our goal was to say, I want to stop the trial when I know the treatment benefit is less than delta, then you would have reached that conclusion at the third analysis. On the other hand, if you want to prove definitely a benefit, then here again, I've listed 3 examinations of data, the estimate and its confidence interval, adjusted for the fact we're repeatedly testing the data. First analysis does not exclude 0. The second analysis does not exclude 0, but the third one does. So there you could argue to reject the null hypothesis of no difference and perhaps terminate the trial. Another complex and agonizing problem is, do you use the same degree of evidence to assess benefit versus harm? There's certainly ways to have symmetric boundaries using group sequential boundaries methodology, which says I want the same level of evidence for benefit or harm, or if you're comparing 2 active therapies, you don't care which direction, but you want the same degree of evidence. On the other hand, how about asymmetric boundaries? In the situation, we have a new treatment versus a standard. You may be interested in having less evidence for a harmful trend than for a positive or beneficial trend. On the other hand, if the new treatment is not proven, but already in practice, as is the case, for example, in the use of hormone replacement therapy, you may want to require more evidence than normal, or at least the same level of evidence as for benefit, because unless you're convincing, you're never going to discourage people to stop a practice. So depending on the situation, a brand new drug, maybe you have asymmetric evidence, less evidence for harm than benefit, but the therapy is already out there and established and in use, but yet not proven. Maybe you need the same level of evidence to show harm than you would for benefit. These issues are discussed in an article that I co-authored with Desmond Julian and Stuart Pocock in 1999. There is no right answer, but it's something you need to consider. What I've plotted here is some statistical methodology that helps you in thinking about this. The top graph and the bottom graph are the O'Brien-Fleming boundaries, as we discussed before. However, the boundary in the middle is a boundary which we've not talked about in detail, but it's a boundary which, if you cross that boundary, you know that you have little chance or less chance of establishing benefit. The lower boundary means that you've established pretty convincingly harm. The upper boundary is that you've established benefit. Let me show you how this would play out in an actual trial. This is a trial called PROMISE. It was examining a drug called milrinone versus placebo in a population of heart failure patients. The graph is the same as in the previous slide, but here I plotted the data from this trial as it emerged over the course of the trial. The stars are below the line of z equals 0, indicating it is in a harmful direction. At some point, the data crossed the middle boundary, suggesting to us that it's unlikely this trial would be positive, claiming benefit. But the trial, as you observe, continued. This drug had a property of making patients exercise better, feel better, and improve various cardiac function. Well, if the mortality had settled down to be no different, as it did, for example, in the Coronary Drug Project, it would still be a useful drug, just not quite as useful as hoped for. On the other hand, if it's harmful, that's a different matter. This trial, in fact, did cross or approach the lower boundary and was stopped, indicating that this drug was harmful in this population of patients. But these statistical methods give you some guidance as to when you are in a point where you're not going to be able to show benefit and where you've established harm. I'm now going to briefly talk about a second analysis method called stochastic curtailed sampling, or simply conditional power. What it computes is the probability or likelihood of a reversal in the trend in the emerging data. You can stop trials early because it doesn't look like you're going to make benefit or harm. When you do that, you of course increase your type 1 and type 2 errors, and I won't go through the mathematical details, but there's an inflation of both your type 1 error and your type 2 error. I've written some formulas here only to provide an opportunity to talk about what this method is about. Typically, you have a test statistic, Z sub capital T, at the end of the trial when the trial is over. You have a current statistic, Z of T, it's whatever the results are on the standardized scale now. You have a rejection region, R, and you have an acceptance region, R bar. The way things normally work is the probability of being in the rejection region on a null hypothesis is alpha, your type 1 error or false positive rate. Your probability of being in the acceptance region, given that the alternative is true, is a type 2 error, or beta. Of course, 1 minus that is power. For conditional power, we modify this calculation slightly. For example, in the first line, we indicate that we would compute the probability of being in the rejection region at the end of the trial under the null hypothesis as we computed before, but now an additional piece of evidence, the current test statistic, is taken into account. I call that probability gamma sub naught. Likely, we would also compute the probability of being in the acceptance region under the alternative hypothesis, given our current data, to be gamma sub A, the alternative. Well, these two probabilities are important because they dictate to us how much we would inflate our type 1 and type 2 error, but the formula is at the bottom of the page. We would compute the first line if we had a positive trend, if things are looking really good, and we want to know if we stop now, how much greater chance of a false positive be taken if we just let it ride to the end. The second line actually is the most useful and interesting, is if we have a negative trend, what's the probability of failing to find a positive benefit given this current negative trend? These two probabilities help us decide whether we should continue or not. The second line is most often used to evaluate futility. Is it hopeless? Things are so negative in the wrong direction that it's hopeless to recover and become beneficial. This probability, this concept, is what generated that middle line of the previous figure. Example, if you were to look at a gamma sub naught of 0.70, the type 1 error would be inflated from 0.05 to 0.071. If you compute that probability given the current trend, be 0.80, you would inflate the type 1 error less because you're more sure of yourself. 0.85, you inflate it from 0.05 to 0.059 because you're even more sure of yourself. If you insist that the gamma naught or even the gamma sub A are 1, then you have certainty, and we call that curtailment. You don't stop until you know absolutely for sure that you can't win or you can't lose. Again, I've computed a formula here for you. It's not important that you understand where the formula comes from, but I present it to only illustrate one point, that this is a very simple calculation to make. The last line is something that you can do almost on the back of an envelope. The key terms are T, the information fraction, the Z sub alpha, which is the criteria you're going to use, say from an O'Brien-Fleming boundary or just even a conventional boundary. For 0.05, that would be, say, 1.96. And then the Z sub I, the test statistic that you have at this moment. And there's a theta sitting in the middle of that formula. Theta is either the null hypothesis of no difference, or it's the hypothesis that you assumed in your alternative. Phi is the normal density function that you can look up in the back of almost any statistical textbook, or almost any computer software program with a statistical package will have this function in it. So it's a very simple calculation. That's the main message I want to leave with you. What's going on sort of visually is that you, again, on 0 to 1, the information fraction, the B of T is just the test statistic times the square root of T. So it's on this scale, so I can plot it simply. You're at some point T in the trial, you look at where you are, and you make some calculations about where you'd be if you continued. You project what the test statistic would look like. Under the null hypothesis, it's not going to get any better. Under the current trend, it's going to continue to where you have the value B of 1 at the end. So you're able to project where you'll be at the end of the trial, given that things stay the same, if they're better or worse. Again, I'm going to go through these relatively quickly. The details are not so important, but simply to say that whether you're measuring survival or success and failure as a binomial outcome, or you're measuring mean response, that theta parameter is relatively simple. For example, in survival, it's the number of deaths you expected in your design, modified by the log of the hazard ratio, which you'd get from any statistical package. The binomial outcome would be the difference in the proportions that you expected in your study and the number of patients you expected to randomize. Again, simple calculations. This is the value for differences in means. The assumed mean difference, the variability sigma, or the standard deviation sigma, and the number of patients you expected to randomize. The details are not so important as the idea that the components of this are things you already know, are really simple, and which makes this whole computational aspects of conditional power quite doable. In the BHAT trial, for example, which I indicated the emerging data, when it came to the point the trial was going to stop, there were 183 deaths on placebo and 135 on propranolol. We had an observed lead benefit for the drug. We had a total of 318 deaths. We expected 398, so we were 80% of the way through the trial, 318 divided by 398. The test statistic was 2.82. So under the null hypothesis that there was really no difference in propranolol versus placebo, I can compute the conditional power quite simply to be 0.89. That is, the chances of being successful, even if the rest of the way the therapy is not beneficial, is still almost 90%. So very little chance of not having the same answer if we continued or we stopped now. I've plotted this for alternative numbers of deaths, and you can see the probability remains quite high, and the effect on the type 1 error is quite modest. Let me share with you a couple examples before we close. This is the PROMISE trial. You saw the data briefly before. As I indicated, it's a drug that's used to test its effect on mortality in class 3 and 4 heart failure patients. It was known to affect cardiac function and contractility. A multi-center, randomized, double-blind, placebo-controlled trial with mortality as the primary outcome. 1,088 patients were randomized, followed for a median of 6 months. 90% compliance, which is pretty good. Not one patient was lost to follow-up, which is remarkable. As I said, the trial was stopped 5 months early because there was an increased excess deaths on milrinone compared to placebo. And I've indicated here the two Kaplan-Meier curves that appeared in the publication of this study. The mortality rate on placebo is better than that on milrinone. And here I've plotted the data as before for the sequential boundaries, crossing the boundary into the trial, suggesting harm. As I indicated, PROMISE had an interesting discussion for the Data Monitoring Committee. There was this agonizing negative trend. It raised the issue of symmetric or asymmetric group sequential boundaries. As indicated previously, it crossed the zone of futility. Milrinone was used in patients with heart failure. We needed to rule out neutrality from harm, because if it was neutral on mortality, it still had other redeeming features. If it was harmful on mortality, it's a different matter. So a very illustrative study. Let me close with a trial that showed a positive benefit. The MERIT study was again a trial in heart failure patients. It was among the first studies to evaluate a beta blocker, in this case metoprolol, versus placebo in a heart failure population. It had a run-in period, placebo run-in. It entered 3,991 patients, and it was terminated early with a mean follow-up of approximately 1 year. I've indicated here the baseline characteristics, just to show that there was comparability in key risk factors of age, gender, heart classification, class, and ejection fraction. I've indicated here in this figure the mortality curves for the MERIT study comparing placebo to metoprolol. You'll observe that placebo mortality rate is much higher than the metoprolol-treated patients. In fact, there's a 34% reduction. If you compute a naive p-value, just using the test statistic, forgetting about the fact that you looked at this data several times, the p-value would be an impressive 0.0009. On the other hand, if you adjust for the fact that you had a sequential boundary in place, the p-value would be still impressive, but only 0.0062. The boundaries that were used to decide when the evidence was sufficient to establish benefit is as shown in this figure. It's different than the ones I've graphed previously. In fact, the upper boundary is a variation of the Peto boundary. It's flat until almost the very end. For the upper boundary, this boundary is asymmetric. The lower boundary is not the same as the upper boundary. So different evidence for harm than benefit. In this study, of course, we didn't go in the negative direction. We see a positive benefit emerging very quickly. The first formal analysis took place when 25% of the data were in, and the z-value at that point was 2.550. Didn't cross the boundary at that point in time. It continued on until 50% of the data of the number of deaths were observed from what was expected. At that point, the z-value is a positive 3.807. At that point in time, the monitoring committee recommended terminating the trial for benefit. There was another condition, however, I need to mention. Because a beta blocker in heart failure patients was initially viewed to be a harmful practice, investigators wanted to make sure that they had enough exposure to this drug that, in fact, it would be safe. So the agreement was that we would not stop until we had 50% of the exposure that was expected to the drug. And when we achieved a statistical test statistic and the exposure at the same time, all the things lined up and the committee had met the criteria that was established. If you look at the confidence intervals for the various outcomes, total mortality, cardiovascular mortality, sudden death, or death from heart failure, they all are quite a bit less than 1, indicating metoprolol, the beta blocker, was beneficial. The length of the line indicates the confidence interval. The symbol represents the estimate. Notice the consistency across outcomes. I plotted here the confidence intervals of benefit, the risk, relative risk, and its confidence intervals for classical cardiac risk factors for heart failure. Again, notice the consistency that the estimates are all less than 1. In fact, many of the confidence intervals exclude 1. A rather impressive display of consistency across subgroups that are important risk factors. Well, I've given you a brief tour of the field of monitoring trials for benefit and safety, from the operational aspects of a monitoring committee and the issues that they must deal with and how they might be organized, to the statistical issues that be kept in mind when one repeatedly monitors data. This is not a simple process, but it's quite doable if one keeps in mind these basic principles which I've shared with you. Also to remind you that these principles and these statistical methods are relevant whether you have a larger multi-center trial or your own trial in your own center. The issues are still there. Methodologies still are relevant. I hope that your experiences in monitoring your trials are successful and that you have an operation in place that gives you confidence that your trials are being properly monitored. It's a requirement, no longer something that's optional. Thank you very much.